channel. So in this video, I'm going to talk about the amino acid catabolism, the breakdown of amino acids. Now remember, when we eat proteins, the meat, the eggs, cereals, we are going to get the proteins into our body. The proteins are going to be broken down inside the stomach, maybe by the help of the trypsin, and also part of it in the small intestine. Alright, of course you know the enzymes, the trypsin, the chymotrypsin, the carboxypeptidase, all those other enzymes are going to help us break down the proteins into amino acids. Now these amino acids can be transported into various tissues, one of them being the muscle tissue. Now let us take for example, in our muscle tissue we have alanine. Now alanine, the structure is that is the structure of alanine. So this is our alanine. Now this alanine can react with an alpha keto acid, right? And the alpha keto acid in this case is going to be the alpha ketoglutarate. So we can react this with the alpha ketoglutarate, which is CH2. That's our alpha ketoglutarate. So alpha ketoglutarate. Right. Now these alanine can react with the alpha ketoglutarate now, and what is going to happen? The alanine is going to remove or is going to release this amino group. It's going to lose this amino group to alpha ketoglutarate, and alpha ketoglutarate is going to give the special oxygen to alanine. And this reaction is going to be catalyzed by a particular enzyme, which are just going to see immediately. Now, when we react these two molecules, and these reactions are reversible, so you can see either of them. You can start with the products, then you're going to get what you're now seeing there. So, when we react these two, and this process is going to be called transamination. We're going to call it transamination. Transamination simply means the transfer of an amino group to an alpha ketoglutarate or alpha keto acid to form another amino acid. Now, when we transfer the amino group, which is the first process in the catabolism of amino acid, the loss of the alpha amino group, then we're going to exchange that one. The enzyme that is going to be involved in this reaction is going to do something which is very, very, very nice. Now see this. This enzyme. Now let's say this side we have the alpha ketoglutarate, and this side we have our alanine. Now what is special about this alanine is that it has the amino group, the H3. Then what is special about the alpha ketoglutarate is that it has the special carbon. Now, this enzyme has something called pyridoxal phosphate, PLP. The pyridoxal phosphate is a coenzyme derived from vitamin B6, that is the pyridoxine. Now, the name of this particular enzyme is going to be called alanine aminotransferase. Alanine aminotransferase. So what the pyridoxal phosphate is going to do is that it's going to take the amino group, then transfer it to the alpha ketoglutarate, then also taking the oxygen, then transferring it to alanine. Then so what are we going to have? Now before we get into that, the pyridoxal phosphate is bound to the alanine aminotransferase via the strip base linkage, right? So you know the example of a strip base linkage. You remember from your organic chemistry, the imine was N can be mean divided to CH3, right? So that's an example of a strip base linkage. So this is how the pyridoxal phosphate is bound to the alanine aminotransferase or alanine amino transaminase in other words. So what are we going to form from this reaction? This amino is going to be transferred to the alpha ketoglutarate. So let us stop the part. We are going to have CO, that one, CH2, CH2, COO. Then you are going to have the amino, right? Then this side, what are we going to have? We are going to have C, O, these are O. Right, then I'm going to have CH3, then double. Now, you guys remember this from your biochemistry and organic chemistry? This is called pyruvate, right? So this is pyruvate. Pyruvate. Then what about this one? This is called glutamate. So, glutamate. So these are the products of the transformation process from alanine exchanging with the alpha ketoglutarate. So the alpha ketoglutarate is going to transform into glutamate, then alanine is going to transform into pyruvate. Now, what is going to be the fate of the pyruvate? Now remember this, pyruvate could change or transform into acetyl-CoA. Acetyl-CoA. Via the pyruvate dehydrogenase complex, right? The transition step. Right. So the acetyl-CoA are now getting into the TCA by reacting with oxaloacetate from citrate, from isocitrate, alpha ketoglutarate, succinyl-CoA, succinate, fumarate, malate, then oxaloacetate. So that is phase 1 of this the TCA. That is the first phase of pyruvate. Then pyruvate can also be converted into lactic acid, right? So pyruvate can be converted into lactic acid. Lactic acid. And this can involve an enzyme called lactate dehydrogenase. So this enzyme that converts pyruvate into lactic acid is called the lactate dehydrogenase. Lactate dehydrogenase. Whenever you see a dehydrogenase, to mean you are going to have the reducing precursor for the components. So the NAD, the nicotinamide adenine nucleotide, NAD plus is going to be converted into NADH. Alright. What's going to happen to the lactic acid? Lactic acid can be transported into the bloodstream, then into the liver. So we have our lactic acid. Now lactic acid can be converted again to pyruvate. Right? Now, the name of the enzyme still remains the same. The lactate dehydrogenase is also going to convert lactic acid into pyruvate. So we have lactate dehydrogenase. Lactate dehydrogenase. Then, what are the reactions? We are going to have NADH is going to be converted into NAD plus. It means it's going to be oxidized. So we are going to reduce lactic acid into pyruvate. Alright. Then what's going to happen to pyruvate? Pyruvate can undergo gluconeogenesis. Remember, gluconeogenesis was the process by which we synthesize glucose from non-carbohydrate precursors. The lactic acid, the pyruvate. So, with the gluconeogenesis, pyruvate can convert into glucose 6-phosphate. Right? So let me just write here gluconeogenesis. So this is gluconeogenesis. Then glucose 6-phosphate can be acted upon by an enzyme called glucose 6-phosphatase, and it's going to convert this one into glucose. So this enzyme is going to be called glucose 6-phosphatase. Glucose 6-phosphatase. It's going to release the phosphate group. Then you're going to have our glucose. Then glucose can maybe be transported back into the muscle, maybe to provide energy needed for particular reactions there. So this is the Cori cycle. If you know the Cori cycle, these are the Cori cycle. So let me just write here Cori cycle. These are maybe and be transported back into the bloodstream, then into the muscle. Alright, now what is the fate of our glutamate? That is the fate of the pyruvate. So our glutamate will be transported to the liver, right? Let me just show what is going to happen to our glutamate. It's going to be transported to the liver, right? So we have our glutamate in the liver. So we have CH2, right, that's our glutamate in the liver. Now what is going to happen to this glutamate? This glutamate is going to be acted upon by an enzyme called glutamate dehydrogenase. So let us see. It's going to be acted upon by an enzyme called glutamate dehydrogenase. Glutamate dehydrogenase. Now, whenever you see a dehydrogenase, you know we are going to involve the reducing components. So we are going to have the NADPH, NADP plus is going to be converted into NADPH. You know the NADPH can be used to in fatty acid synthesis, you can use the NADPH, and also maybe in neutralization of these radicals. You know, radicals can cause a lot of oxidative stress, so you can neutralize them using the NADPH. Right. Now, this reaction is going to involve, so it's a two-step reaction, but we're just going to bypass the first step so that you can add a water molecule onto this. When you add a water molecule, what are the products? The products of this particular reaction are going to be alpha ketoglutarate, and also going to release ammonia. Alright, that's pretty good. I'm going to release ammonia. Now, the synthesis of the breakdown of the glutamate into alpha ketoglutarate and release of ammonia, this process is called oxidative deamination. So this process is going to be called oxidative deamination. Oxidative deamination. We are deaminating our glutamate. This was our glutamate. Alright. Now, we have our alpha ketoglutarate, but let me talk about the ammonia. Ammonia, you know, is very much toxic, and therefore the body will not desire to have it as ammonia. So what is going to happen? The ammonia is going to be transported into the mitochondria, where we are going to run the urea cycle, right? Yeah. Urea cycle comes inside the mitochondria, and we are going to have a special video for the urea cycle so that you are going to see how we can convert this toxic ammonia to its toxic, and therefore the body will try to find a pathway on how to break it down into non-toxic molecules. That is the urea cycle. We'll find a video for that. Then the alpha ketoglutarate can either be taken. You know, alpha ketoglutarate is a component, which is an intermediate in the TCA, right? So we can maybe take it to the TCA, or we can also take it to another transamination reaction. So maybe we have aspartate, that is C. This is the zitarionic form of aspartate. Now, zitarion simply means that the net charge is 0. Meaning you have a positive charge on one side and a negative charge on the other side. So it's called a zitarion. So this is our aspartate. So this alpha ketoglutarate can again come and engage in a transamination reaction with the aspartate. So you can react it with the aspartate. Alright. These are alpha ketoglutarate. Alpha ketoglutarate. So when we add these two, going to form. And remember, these reactions are reversible. So maybe when we see in other books, we start with the products. Maybe, for example, in this case, let us see what we are going to form first. When we react aspartate and alpha-ketoglutarate in the presence of an enzyme, this particular enzyme is going to work almost the same, actually the same way as the alanine aminotransferase. So let us see we have that enzyme here. So we have our aspartate. The aspartate is here. Aspartate. Then you have our alpha-ketoglutarate. Then you have our pyridoxal phosphate right there. Alright. What is unique about aspartate is that it has the amino group. And what is unique about alpha-ketoglutarate is that it has a special oxygen. So what the pyridoxal phosphate is going to do is that it's just going to pick the amino group from the aspartate, then transfer it to alpha-ketoglutarate, then pick the oxygen, then transfer it into aspartate, so that we are going to form two products. Now here are the products. We are going to do or release the amino group. That is always the first step of the catabolism of amino acids, right? Now, aspartate. So what are the products? We are going to have. Now we are going to have C. The aspartate is going to be converted into C O negative, CH2, COO. Then we are going to have the double bond right there. Then this side we are going to have is going to be the amino group. So we are going to have C right there. Right. Then the amino group right here. Alright. Now, what are the names of these products? This one is called glutamate. Just as you saw here, we still have our glutamate right there. Then, what about this one? Remember this from your organic chemistry? Now, this one is going to be called oxaloacetate. Oxaloacetate. Right? We also know it from the TC. It's also one of the intermediates in the tetracarboxylic acid cycle. Now, what is the name of this particular enzyme that catalyzes this transamination reaction? This reaction is called transamination. This particular enzyme, which is bound to the pyridoxal phosphate strict base linkage, is going to be called alanine aminotransferase. Now, aspartate. Aspartate aminotransferase. We're going to catalyze that particular reaction. Now, our glutamate can also go to the same pathway. It can act on it can be acted upon by the glutamate dehydrogenase, can generate ammonia, and so on and so on. So, what is the fate of our oxaloacetate? Now, just as I said, oxaloacetate is a component of the TCA, right? Where we convert pyruvate into acetyl-CoA. Then react this by oxaloacetate and react with the acetyl-CoA to form citrate. Then citrate into isocitrate. Then isocitrate into alpha-ketoglutarate. Then into succinyl-CoA, then into succinate, then to fumarate, then into malate. Then malate can convert into oxaloacetate. Right. That is our TCA, tetracarboxylic acid cycle. Alright. Now, that's where we can now take our oxaloacetate. Can react in the TCA. This is pyruvate. Now, suppose these are mitochondria. So, let us see. Suppose these are mitochondria right there. And you know there is several amino acids that can be shunted into various intermediates of the tricarboxylic acid cycle. Now, so for example, fumarate, phenylalanine can be converted into, let me see different, right? Phenylalanine can be converted into fumarate. Then you can have, for example, the isoleucine can be converted into succinate, and now isoleucine. We can have the alpha ketoglutarate that can be glutamate. Glutamate can be converted to alpha ketoglutarate. We can have aspartate. Aspartate can be converted to oxaloacetate. So various amino acids can be converted to various components of the TCA so that you can generate energy. Because remember, from the TCA, we are going to generate NADHs, we are going to generate also the FADHs, FADH2. We are also going to generate some energy in form of ATP. Now the energy produced from the TCA is going to be through the substrate level phosphorylation. So that is energy. Right. And remember, NADHs and FADH2 can also be taken to the electron transport chain. So you can take these to the electron transport chain. Then you can generate energy in form of ATP through oxidative phosphorylation. Right. So you see how we are breaking down these amino acids to generate energy. Now, oxaloacetate can also be used in gluconeogenesis. Now let us see how we can use oxaloacetate in gluconeogenesis. Remember, this reaction here is reversible between oxaloacetate and malate. Now, oxaloacetate can be converted back to malate because oxaloacetate cannot penetrate through the mitochondrial membrane. Now, what is going to happen? We are going to convert oxaloacetate into malate. Then malate can now penetrate and gets out of the mitochondria. So we are our malate. Alright. Then out of the mitochondria we can convert now into oxaloacetate. Then oxaloacetate can be converted into phosphoenol pyruvate. That is PEP. And the enzyme that catalyzes this particular reaction, of course, you know, is going to be called PepCK. We're going to have our PepCK. That is phosphoenol pyruvate carboxykinase. It's going to catalyze and it's going to put some phosphate there. So, phosphoenol pyruvate. Phosphoenol pyruvate can now move up to the gluconeogenesis. To generate glucose 6-phosphate. Then glucose 6-phosphate, of course, you know, can be acted upon by glucose 6-phosphatase. So glucose 6-phosphatase can leave the phosphate group from the glucose 6-phosphate and convert this into glucose. And convert glucose 6-phosphate into glucose. So the enzyme here is going to be called glucose 6-phosphatase. Just like we get in the case of pyruvate. So, glucose 6-phosphatase. Alright, now the process by which we generate glucose from non-carbohydrate precursors is the gluconeogenesis. So that in the same way we can mobilize the oxaloacetate to generate glucose. Now maybe this glucose can also find its way into the bloodstream. Alright, that is pretty cool. Now, that is it we have for amino acid catabolism. But before we finish, we're going to have now when we have alanine aminotransferase and the aspartate aminotransferase, when, for example, you are doing some diagnostics, you are running a blood test, you may find the elevated concentration of alanine aminotransferase in blood. And also, we're going to have the aspartate. We can also find high levels of aspartate aminotransferase in blood. Now, when you run a blood test and you find the presence of these particular enzymes, that is to say that you have defects or disorders in those particular organs. We know the ALT and the AST, or the alanine aminotransferase and the aspartate aminotransferase, are contained in the muscle, for example, in the liver, in the heart. So when we find them in blood, that is to say that we have defects in those particular organs, and therefore the presence of these particular enzymes in the blood is going to show or help us diagnose infections such as the myocardial infarction. So myocardial infarction, that's a disorder in the heart. Then another one, we're also going to have maybe this is a liver disease. So liver disease, and also in the muscle, maybe muscle dystrophy, or there's a disorder in the muscle. So you can talk of muscle dystrophy. So these are the disorders that can be associated by the presence of ALT and AST in blood, or maybe in serum, or even in plasma. Alright. Another one, we can also talk of the disorders associated with the amino acid metabolism. So let us write them here. Disorders associated with the amino acid metabolism. So number one, we can have diseases like homocystinuria. Now homocystinuria. Homocystinuria. Right? We can also have alkaptonuria. Alkaptonuria. We can also have maple syrup urine disease. Maple syrup urine disease. Alright. Then we can also have albinism. Albinism. When you have a defect in the tyrosine metabolism, and there is a deficiency on tyrosinase enzyme. Alright, what is the meaning about the disorders? We have also have another one called phenylketonuria. And this is a defect in the phenyl hydroxylase, or phenylalanine hydroxylase. And we're going to see how that's going to affect that. Now, those are the disorders associated with the amino acid metabolism. You also have the presence of ALT and AST in blood. So that is all that we have for the amino acid metabolism, guys. And in our next video, we are going to see how we are going to shunt this ammonia we produced here, and of course we know it is toxic, into the mitochondrion, so that you can run the urea cycle, so that you can get rid of the toxic ammonia to get it into non-toxic or harmless urea. Thank you very much, and if you guys found this video helpful, please make sure to subscribe to my YouTube channel, hit that like button and comment. Let's meet in our next video. Bye.